Hey guys, guess who's back? Yep, it's your favorite biology teacher. Hi. Back with Standard 4. Standard 4 covers the evolutionary theory. And Standard 4 has a lot in it, so I want to do this in two parts. So the first part, we're just going to talk about, I think it's 4.1, and 4.1 is talking the difference between chemical and organic evolution. I'm going to talk about 4.2, which is the evidence for common ancestry and evolution. And I will talk about 4.5 and 4.6. 4.5 is going to be Darwin's theory, and 4.6 are mechanisms of speciation. I'll do a separate video where I actually look at, 4.4 is in this as well. I'll do a separate video where I actually look at 4.3, which is cladograms and phylogenetic trees. Okay? Alright, so let's go ahead and get this popping. So first things first, evolution literally means a change over a period of time. And so when we're looking at evolutionary changes, it's important to know that we are just looking at how things have changed over a period of time and also the evidence that supports that theory of evolution. Now, the cell theory addresses a couple of things. It tells us what cells are, and cells are the basic unit of structure and function for all living things. The cell theory tells us where we can find cells because we know that all living things are made of one or more cells. And the cell theory tells us that cells come from pre-existing cells. But what the cell theory does not address is where did the first cell come from. According to the cell theory, cells come from other cells. Okay, well, where did that actual first cell come from if cells are coming from other cells? So, in order to address that concept, we have to look at the different types of evolution, which are chemical evolution versus organic evolution. Now, chemical evolution basically explains how we go from having elements, so carbon, hydrogen, oxygen, phosphorus, nitrogen, those elements of the periodic tables, those are your chemicals. How do we get from actual inorganic things, from elements to these organic compounds and ultimately the first cell? So that's what we're looking at with chemical evolution. How do we get from elements to major organic compounds to the actual first cell? Now we know that scientists believe the first cells on Earth were prokaryotic, they would be simple cells, heterotrophic, meaning they had to consume nutrients in order to get energy, and anaerobic because early Earth's atmosphere is believed to have been an anaerobic atmosphere. We believe Earth is about 4.6 billion years. And so chemical years old, so chemical evolution would have happened in the beginning around 4.6 billion years ago. Now, there has been an experiment. So we had the Oparin-Haldane hypothesis, which hypothesized how this happened, and then Miller and Urey actually performed an experiment. They created this apparatus that simulated Earth's early conditions. So they had, they used electricity to simulate lightning, and they had different elements in there, but they did not put oxygen because remember, we believe that early Earth's atmosphere was anaerobic. And in this experiment, they were actually able to make amino acids. Now, there was another experiment that was actually able to produce protocells made by different scientists, and protocells actually behave similar to the plasma membrane, where in an aqueous environment, they actually arrange into this bilayer. So, scientists have been able to produce amino acids, which is a major organic compound, monomer, and also protocells, but have not yet been able to produce an actual cell in a laboratory from just these elements. So it is believed that chemical evolution happened in the beginning and it is done with, and that is how we got from having these elements to the actual first cell, which is believed to have been a prokaryotic heterotrophic anaerobe. Now, organic evolution picks up where chemical evolution leaves off. Organic evolution tries to explain how do we get from this first cell, this prokaryotic heterotrophic anaerobe, up to these eukaryotic autotrophic cells, and they're not only just eukaryotic autotrophic cells, they actually combine to make these multicellular organisms, and then among these multicellular organisms there's so much diversity. So organic evolution is trying to explain the diversity and also the unity within that diversity. The evidence that supports organic evolution, one of the things that we look at is called the endosymbiotic theory. Now, the endosymbiotic theory is a theory that seeks to explain the existence of mitochondria and chloroplasts. Mitochondria and chloroplasts, if you recall, are our energy organelles, where chloroplast is an energy transformer. It transforms radiant or light energy into chemical energy and stores it in the bonds of carbohydrates. And then mitochondria is able to efficiently break down those nutrients and transfer that energy from the bonds of the nutrients into the bonds of adenosine triphosphate, or ATP, which is simply a modified RNA nucleotide. Yes, I am reviewing those things as well. All right, but anyway, the endosymbiotic theory is basically a theory that tries to explain the existence of these two cell organelles. And so basically what it's saying is that an ancient cell engulfed a prokaryotic cell. We believe purple sulfur bacteria was the first cell to have been engulfed by this ancient cell, because remember, the first cells would have been heterotrophic, so they would have had to consume things in order to get their energy. But instead of actually breaking it down, they developed a symbiotic relationship, living and working together, called endosymbiosis, because something went inside endo of something else, and they began to work together. And those cells became the precursors for our eukaryotic heterotrophs, and they would contain mitochondria. Now, some of those cells would have gone on to become our eukaryotic heterotrophs. So you have your fungi, you have your animalia, you have some of your protista. But then other cells that already contain this relationship, that already have this relationship, are believed to have engulfed a second bacteria called cyanobacteria. And cyanobacteria was autotrophic. And so they also developed this symbiotic relationship, and these cells were the precursors to our eukaryotic autotrophs. So with this theory, what scientists believe is that mitochondria evolved prior to chloroplasts, and so therefore our eukaryotic heterotrophs do not have chloroplasts in them, and only our eukaryotic autotrophs, which would be our plants, would have that chloroplast organelle. But notice they would have both mitochondria and chloroplasts, which we know is true because our eukaryotic heterotrophs can do aerobic cellular respiration, whereas our eukaryotic autotrophs do both photosynthesis and aerobic cellular respiration. Now, there is evidence to support the endosymbiotic theory, and there are 4 pieces of evidence that support that. Number 1, both mitochondria and chloroplasts are double membrane enclosed organelles. There's a cell within a cell, 2 membranes. They're double membrane enclosed. The other thing that's unique about mitochondria and chloroplasts, we talk about 2 and 3 at the same time, is mitochondria and chloroplasts have their own DNA and their own ribosomes. What is unique about this DNA and the ribosomes, the DNA is in the form of a single circular chromosome. Where else do we see circular chromosomes? Prokaryotes. So if purple sulfur bacteria or cyanobacteria was engulfed, they would have had a single circular chromosome because prokaryotes have a single circular chromosome. Now, the thing about the ribosomes is ribosomes come in different sizes. Prokaryotes have smaller ribosomes than eukaryotes. The ribosomes that we see inside of mitochondria and chloroplasts are the same size ribosomes we see inside of bacteria. Now, this is important because mitochondria and chloroplasts have a bunch of proteins that are used and enzymes that are used in their processes, but they are able to make their own proteins because they have their own DNA and they use their own ribosomes to make their own proteins. They don't have to get instructions from the nucleus on what proteins to make and when to make them. The last piece of evidence, that 4th piece of evidence, is mitochondria and chloroplasts also divide independently of the cell cycle. All other organelles are copied or reproduced during the cell cycle. When a cell is actively dividing, as it prepares to divide its nuclear contents, it also makes more of the organelles that will be needed in the new cell. Mitochondria and chloroplasts, however, are able to divide independently of the cell cycle, and they do so through the process known as binary fission. What else do we know divides through binary fission? Prokaryotes. So, this is the evidence that supports the endosymbiotic theory when mitochondria and chloroplasts were developed through a cell engulfing another cell and developing a symbiotic relationship. Now, So that is like all the precursor stuff. So this is how we think we came about, how we got from elements to the first cell, which would have been a prokaryotic heterotrophic autotroph, and then how this first cell would have ultimately ended up, this prokaryotic heterotrophic anaerobic cell ultimately turned into these eukaryotic heterotrophic cells or eukaryotic autotrophic cells. So how do we get there? Now, the next thing is, okay, great, we have these different types of cells, but we talk about how evolution explains the unity and diversity amongst all living things. So, what's the unity? All living things use the same genetic code. We have the same 4 bases in DNA. Adenine, thymine, guanine, and cytosine are found in DNA, and then RNA or other nucleic acid uses adenine, uracil, guanine, and cytosine. The base pairing, adenine pairs with thymine in DNA, adenine pairs with uracil in RNA. Guanine and cytosine will always pair with each other. You have your pyrimidines versus your purines, and then one pyrimidine pairs with another purine. This is the same from organism to organism. Also, DNA is transcribed to RNA, and RNA is translated to proteins, and those proteins are the things that's going to be expressed as your traits or have your enzymatic functions. From organism to organism to organism, this is the same. So there is unity amongst organisms, but there's also diversity because it is the order and arrangement of those nucleotides, those nitrogenous bases, what order they come in, how many you have. That is what distinguishes one organism from the other one. What proteins are being made and how they are being expressed. So, the evolutionary theory, of course, scientists were curious, like how do we have all these different things? And the evolutionary theory took some time to develop. We know that Charles Darwin is known as the father of evolution. We also know another scientist by the name of Alfred Wallace came up with a similar theory of evolution through natural selection, but due to other circumstances, Charles Darwin was able to publish. He published a paper with Wallace, and then later on he published On the Origin of Species, which gained a lot of recognition, and he became ultimately known as the father of evolution. Now, Darwin did not set off to do this initially, and he did study the work of other scientists, and it is a combined work that ultimately led to this theory of evolution through natural selection. The key to natural selection is there must be genetic variety within a population of organisms. And we know that a population is a group of organisms, the same species, within the same space at the same time. And in order to be classified as being of the same species, you must be able to produce fertile offspring, which means you must be able to have babies that can make their own babies. Not every member of the population has to be able to have babies, but as a whole, the species must be able to reproduce in order to be considered, the organisms be able to reproduce in order to be considered of the same species. Now, one of the components of Darwin's theory of evolution is that there is a common ancestor. Okay, there is a common ancestor amongst organisms. He is not the first scientist to come up with the theory of common ancestry. In fact, Jean-Baptiste de Lamarck also believed in a common ancestor, but one of the things that made his theory not believable was he also believed in something called use or disuse, which basically it was use your traits or lose them. The animal that we talk about a lot with Lamarck is his theory on giraffe necks and how they went from being short necks to long necks was simply because they stretched their necks because the plants were getting taller. And if you stretched your neck while you were alive, when you had babies, then they were born with longer necks. So he wasn't on the mark with that part, but he did believe in a common ancestor. So we often say Lamarck wasn't on the mark. Another part of Darwin's theory of evolution through natural selection has to do with something we often call survival of the fittest. Now, being fit in nature, in this term, is not considered like being buff or exercise. It means you're best suited to survive. And just because a trait is fit now doesn't mean it will always be, okay, most fit. So basically, survival of the fittest has to do with looking at the increasing competition and how having that genetic variation is required, and the organisms that have the best traits are the ones that actually survive to reproduce. Competition would increase due to the population increasing and competing for resources. And so if the environment changes, so the organisms that have the best trait that are suited for that environment tend to be the organisms that survive to reproduce. They also tend to have more babies than they can support, because the more babies I have, the greater the chance of the genes being passed on into the next generation. So the goal is to keep the genes in the gene pool. Now, the only way that evolution can happen is if there's already variation within the population. So we often look at this. We can use, we can show how natural selection can actually drive populations. Normally in a population, we see like a bell curve where we have, so this would be extreme 1, over here is extreme 2, and here's going to be my average trait, okay? So most of the time, most organisms tend to fall somewhere within that average range. But notice I had to have variation within this population, right? So let's say this is very short, this is average height, this is very tall. So some organisms are going to be very, very short, some are going to be very, very tall, but most of them are going to fall within that average range. So that's what we mean by a bell curve. Now, natural selection can actually drive a population one way or the other, or in different directions. So there's 3 different ways natural selection can influence a population. Sometimes we see where the favorable trait is the average trait. So what we see is a decrease in the extremes and an increase in the average. This is what we call stabilizing, okay? This is a stabilizing drive because it's going to stabilize towards that average trait. Now, other times, natural selection can actually drive a trait towards one extreme or the other one. So in this case, it's going to select against the average, selects against one of the extremes, and then favor the other extreme. So this is called directional. Now, directional can go one way or the other one. I just chose to go towards extreme 2, but I could have gone towards extreme 1. Now, that last type of way that natural selection can influence a population is when it selects against the average. So one extreme or the other one is good, but it's bad to be in the average range. And if it selects for both extremes but against the average, then we call this disruptive, okay? So there's 3 ways that natural selection can actually influence a population. It can stabilize it, it can push directional, so one extreme or the other, or it can be disruptive where it selects for both extremes. Now, this is all can only happen if there's already variation, genetic variation within the population, and the organisms that are best suited for that environment are going to be the ones that are going to live to reproduce and pass on their genes. So you're going to see a shift in the allele frequencies, which will then influence the phenotypes within that population. And so the next part of his theory was that there will be descent with modification, which means from one generation to the next, you're going to see some allele frequencies variations. Okay, there's going to be variations from one generation to the next. And so the key thing, though, to know is that when it comes to evolution, is that populations evolve. Individuals do not evolve. Populations, okay, that group of species living within an area at the same time, their allele frequencies, the frequency of an allele in the population can change, which can lead to evolution. So populations evolve, not individuals within that population. The evidence that supports the evolutionary theory, we can look at comparative anatomy, okay? Comparative anatomy is when you compare the anatomies of different organisms. The 2 that we tend to look at are homologous structures and embryological similarities. Homologous structures, if you were to look at a human's arm or a bat's wing or a cat's leg, right? You're going to see the same structures of bones in the same type of location. You have the ulna, the radius, the humerus, the ulna, the radius, the carpals, the metacarpals, but they're used for different things. So there's going to be similar structures but different uses. So those are called homologous structures, and that suggests that there was a common ancestor. The other things, if you look at embryos, during early development, embryos look very similar. You can't tell one from the other one. So basically you have gametes which come together to form a zygote. The zygote develops into an embryo, the embryo becomes a fetus. So we're looking at the embryo stage. So when there are embryos, you can't tell a tortoise from a human from a pig, but as it develops into a fetus, then you begin to see the differences. But the embryos are going to be very similar. You can look at the fossil record, which is the position of, you can see the fossil record actually favors vertebrates, so it's incomplete. If you look at their location within the earth, you will see that older fossils are at the bottom, or you can do radiometric dating where you get the half-life of things like carbon-14. Molecular and biochemical similarities, different organisms will have more gene sequences that are similar or protein sequences that are similar. The more they have in common, the more closely related they are. So you can look at the percentages of gene sequences that are similar between organisms, or you can look at the amino acid composition of different proteins. And remember, the more you have in common, the more closely related you are. And also biogeographical distribution, where you see similar fossils in different locations. And those different locations can be like, you know, Africa versus Asia, where you see similar fossils. Now, when it comes to all of that, we have other sciences. So we talked about Lamarck. Mountain studied population, so he predicted the struggle for existence because of overpopulation. Lyell is the scientist that actually looked at how Earth processes, how Earth has changed over time, and how that would contribute to changes amongst the different species. So those are the other scientists, and Darwin actually looked at their work to help him come up with this theory of evolution through natural selection. Now, the last thing we're going to talk about in this video is speciation. And speciation is when organisms become different species. We normally see this through what's called divergent evolution, where one population is somehow separated or isolated from each other. They develop into different populations that even if you put them back together, they would no longer be able to produce fertile offspring. So therefore, they are classified as different species. So that's why species is, or must be able to produce fertile offspring, okay? So not like breeds of dogs, right? Those are breeds, not different species. We know that a poodle and a bulldog can still have puppies. Even though they're different breeds, they can still produce offspring, and then those puppies can have more puppies. They can produce fertile offspring. But think about a horse and a donkey. You can breed a horse with a donkey and make a mule, but mules are infertile. They can't make more mules. The only way to get mules is to breed a horse with a donkey. So horses and donkeys are not of the same species. Even though they can have a baby, their babies can't have babies. So speciation is usually caused by geographic isolations, where populations end up separated by some type of geographic, like a river or mountain range, or something happens that separates members of the population, and then after several generations, because of the alleles that were available within that population, they develop into different species. And so even if you put them back together, they will not produce fertile offspring. So therefore, they're now classified as different species. The other type of isolation is called reproductive isolation. Now, reproductive isolations can be prezygotic, meaning the egg is never fertilized, or postzygotic, meaning the egg is actually fertilized to form that zygote. Your prezygotic examples would be temporal, so the timing's off. I'm diurnal, you're nocturnal. I breed in the spring, you breed in the summer. I live on water, you live in land. Well, that would be not temporal, that's geographical. But temporal, the timing's off, okay? We don't breed at the same time, so because we don't breed at the same time, we'll never actually mate. Behavioral, different behavior practices. I may scream, you may sing, right? That's your mating call. We don't recognize each other's mating calls, so we never actually mate. Or mechanical, the parts don't fit. Postzygotic means that the egg is actually fertilized, so a zygote is formed. Your postzygotic barriers can either be a hybrid breakdown, inviability of the zygote, or you can have infertility of the hybrid. So inviability basically means the egg was fertilized, but it terminates before a baby is born. Hybrid breakdown, the egg develops, the baby is born, it's very sick or weak and usually dies young before it can actually have the chance to reproduce. Or hybrid infertility is like with our mule. Even though the baby is born and the baby lives and survives, they're infertile and cannot produce their own offspring. All right, guys, that was the first part of Standard 4 on evolution. Toodles.